Hi, I'm Bridget.
Hi, I'm Brittany. In this lab, we use the RNAi system plus gene silencing to change and manipulate a worm's morphology.
On the first day of lab, we received wild-type C. elegans that we would observe in their natural state.
As you can see, they move in an S shape throughout the plate. There are 6 stages of life. Here you see an egg, which is the first stage of life. Here you can see an L1. It is the second stage of life. It is the smallest worm. Seen here is an L2 worm. Next we have an L3 worm. This is the fourth stage of life. This is an L4 worm. It looks very similar to the adult worm. This is an adult worm, which is the last stage of life.
The next day, we transferred the wild-type C. elegans from the starter plate with OP50 bacteria on NGM light agar to the feeder plate with the dsRNA on NGM light and IPTG.
By chunking, we transferred the C. elegans from the starter plate to the feeder plate, which had the double-stranded RNA in the bacteria.
We had the DPY11 gene. This gene had an impact on the growth hormone in the worms. This caused them to be short and fat. The gene affected ray morphology and body shape.
After 5 days, we went back to observe the changes. Our plate didn't show which change, it only appeared in a couple of worms. We observed other plates from other periods that had our gene. Those plates showed more change.
They became shorter and fatter. They became so short you could mistake an adult worm for an L2 worm.
Many people are probably wondering how this process works. How did we change the shape of our worms? This is how.
So if DNA is locked in the nucleus, how do we get the information out to build our creature? Well, that's what RNA does. That scribe, copying recipes out of the cookbook and throwing them out the window, out to the cytoplasm C that makes up most of the cell, all those recipes floating through the air, they are RNA. And to finish up, in that C, you see hundreds of thousands of, well, we've made them into little guys with chef hats. Lands on a chef. Whatever it is, he cooks it.
Whatever it is, he cooks it.
And each recipe is for a protein. These chefs are basically building you. You are made of proteins. And because of RNA, we can copy, we can distribute, and we can cook up you and me. Cells from time immemorial have had a mortal enemy called the virus. So let's imagine that the virus is a pirate ship. It lands. It then sends invaders inside the cell to shower recipes down to those cooks. But some of those recipes, you'll notice, look a little different. And what's in these recipes is not good for the cell. So the theory is that long ago, cells developed a secret defense system which we will call the cop. What the cop does is when viruses invade and create showers of murderous recipes, the cop looks and thinks, some of these have a very fishy shape. It's a chemical difference which comes down to some of the viral recipes are 2 pages instead of 1, and one side is a mirror image of the other. But the point is, to the cop, there's something not right about this shape. And the cop destroys the recipe. Destroys not only the oddly shaped version, whenever he sees that recipe, oddly shaped, regular shape, that recipe in any form must be destroyed to defeat the virus.
This lab was very interesting for both of us because we got to see how changing one gene affected the whole body of a